Hi guys, this is the final instalment of Biology with Miss Cherry for Module 2. Thank goodness we finally got here. Proud of you for making it this far if you've managed to and you've kept up, well done. I know it's not the easiest topic to do by yourself at home. So to begin with, we are going to take a recap on things that we've done over the last few lessons and go back to looking at the immune response. So we're going to start by thinking back all the way about 5 lessons ago to lesson 2.16 and 2.17. That's actually 3 or 4 lessons ago, isn't it? So pause now and you're going to have a few minutes to think about what are the 3 ways that white blood cells can fight off infections. And when you've done that, see if you can remember what a phagolysosome is. Now you might want to think about breaking up that word. So pause me now and then come back when you're ready to get the answers. Okay, so hopefully you should have had a little bit of pausing time to think about the 3 ways that white blood cells can fight off infections, and I'm hoping that you will have had the following. We can say that the white blood cell produces antibodies. We can say that the white blood cell is going to undergo phagocytosis, or we can say that the white blood cell is able to produce toxic chemicals which destroy the pathogen, like perforin. Okay, remember that's going to form perforations in the cell membrane, the cell wall, and cause it to burst. Now, what is a phagolysosome? If we think about it and we think back to lesson 2.17, phago means that it's to do with white blood cells and specifically phagocytes. Oh, blood cells. Phagocytes. And the lysosome is the vesicle that contains hydrolytic lysozymes. Ooh, hydrolytic lysozymes. Okay, so the phagolysosome is what you get when you've merged a phagosome, which is the vesicle that engulfs the pathogen or the foreign material that engulfs pathogen, and then that merges with the lysosomes that have the lysozymes in there. Sorry, the spelling of that lysozymes is wrong there. Okay, so not too difficult for starter today. We're going to take it easy today because it's been a long module and a busy week, even if it is only Monday when you're watching this. But, you know, we're all tired. It's the end of module 2, thank goodness. So this final lesson is all about HIV. We're going to take a recap on viral structure and we're going to have a look at how HIV replicates and then how we can treat HIV using antiretroviral medications. So to begin with, structurally, viruses are non-living, and it's a really important point to draw attention to that. They are not prokaryotes. Okay, we know that they're not prokaryotes because they don't have all of the components that make an organism prokaryotic. Okay, they've got a variable type of genetic material and they've got lots of layers that we haven't seen in any other organism. We also know they're not living because they require a host to reproduce. And that's one of the major things that mean that something is alive or not. Okay, so when we're thinking about size as well, the viruses are tiny. We're talking like 200 nanometers, which if you can remember back to our standard form lessons from the beginning of module 2, is 200 times 10 to the power of minus 9 meters. Very, very small. And they have to live inside a host to reproduce. As we said, they're not living, but sometimes the hosts are human cells or animal cells. But sometimes the host can be a bacterium. Okay, and if we've got a bacteria that is or a virus that infects bacteria, there's a specific word for it, and that is called a bacteriophage. So a virus that infects bacteria is a bacterio. And this is all one word, I just can't fit it in the screen. Bacteriophage. Now, viral structures are made of four major components that you can see in that diagram and in that little list at the bottom. But again, the structure is going to vary depending on which virus you're looking at. So we've always got those antigens, the viral proteins on the outside. Okay, sometimes they might be called spike proteins, and you probably have heard a lot about Professor Chris Whitty on the news talking about the spike proteins on COVID. They've got DNA. That's not technically correct. Sometimes they can have RNA as well. It's a variable genetic material. So some of them will have DNA and some of them will have RNA. It just depends on the virus again. And then we've got a capsid and an envelope. And you can see there that the capsid is what encloses the genetic material. And like in a letter, the envelope is around the outside of it. Okay. So here's just a little bit more detail about the structure of the virus. We're not going to spend too long on this. You can pause it if you want a little bit more detail. So as we said, we've got the genetic material, either DNA or RNA, and that is found in the core of the virus. We've got that capsid surrounding the genetic material, which is a protective coat of proteins. We want to make sure that the genetic material is safe. So genes are safe. If they're not safe, then we can probably start damaging our viral protein, and for the virus's sake, that's not very helpful. In terms of detail, you might want to make a note of the fact that capsomeres are the things that make up the capsid, the little individual subunits. The viral envelope is made up of lipoproteins, so that's the combination of a fat and protein. And then the antigens we know are protein molecules, glycoprotein molecules, so protein and a carbohydrate, which are there to specifically recognize T cells, T lymphocytes. Okay, and that's the whole idea of HIV being really dangerous. So the way HIV is transmitted is generally through physical contact. So you have to come into contact with the bodily fluids of an infected person. So that could be through sexual contact, so in terms of passing it through semen, or blood transmission from mother to daughter or mother to son during the womb, mum to baby. As it's non-living, it can't survive outside of the host, so you cannot pick it up from surfaces, so not from indirect contact. Obviously, if you've got a needle on a side that's got blood on it from an infected person, and you come into contact with the blood through it, like a needle stick injury, then there is a chance of infection, but I'm talking about like you can't get it from the surface of a desk that's had some skin on it from someone with HIV. Now, in terms of the virus itself, the HIV virus, it doesn't really have any cellular components that mean that it's going to have to undergo metabolic activity. So there's no respiration going on. There's no photosynthesis or anything like that. There's no protein synthesis. And because of that, because it's basically a dangerous bag of genetic material, it's not going to undergo mitosis. We know that the HIV specifically and other viruses need a host. That's how they reproduce. So HIV itself stands for human immunodeficiency virus. And it's an example of something called a lentivirus. Okay, if you imagine like Lent at Easter is when you stop something for a period of time. And a lentivirus is something that causes a chronic or long-lasting disease after a really long incubation period. Okay, so this, by incubation period, I mean a period of time when symptoms don't appear. Okay, so HIV will just sit in your body and slowly replicate over time until your T cells, which is what it targets, become so damaged, so few, that you get symptoms appearing then. So incubation period is a period of time where the virus replicates but no symptoms. Okay, and as we said down the bottom, it's a chronic infection of the T cells. So we're not talking about the antibodies at the moment, we're just focusing on those T cells. Now, when we're thinking about structure, you can see here that this is a really nice visual of the external surface of the HIV molecule itself. So we're going to take it in turn, we're going to work our way from the outside in of the viral structure, and I'm hoping that this will be nice and clear and you will understand it, and if not, just pop up on the chat or the call or whatever and just ask a question. So on the outside surface, we have got a layer of a lipid bilayer. Okay, so just those phospholipids that we've learnt about previously. Okay, phospholipids arranged with the hydrophobic tails facing inwards and the hydrophilic heads facing outwards. And this is stolen from the host cell. Now what that means is that if it's stolen from the host cell, it's going to contain some self-antigens. Right? And that's a bit of a problem because it means that it can hide from the immune system. Because the body recognises it as just a part of itself, as we've got down there. So it's going to increase the chance of evasion, and it means that we've got an increased chance of ability to infect. So if it can persist in the blood for a little bit longer because it's hidden, then it's just going to make everything a little bit worse. Okay. On the outside of our structure as well, we've got glycoproteins that are from the virus. Okay, they're viral glycoproteins, and they enable docking, so attachment or biological attachment to receptors on the host cell, the target cell, those T cells that we're looking at. Okay, so this is on the viral envelope. We're working outside in, so this is the outermost layer. As we move in slightly to just outside of the capsid, but not quite where the genetic material is, we've got lots and lots of proteins just outside the capsid. And these are there to help with budding, which is the process of viral release from the host cell. And that in turn causes the destruction of host cells. So what we can see happening when we take a look at the viral life cycle in a little bit of time is that the virus itself, when it comes to burst out of the host cell, steals some of that phospholipid bilayer that we've already spoken about and breaks a little hole in the cell. And if that happens too many times, we've got lots of viral particles trying to leave as well, that's going to make too many perforations in the host and kill that host cell. And the RNA, okay, the genetic material of HIV, so HIV genetics are RNA, so not DNA, they're contained inside the capsid, inside those capsomeres, that structure that's made by those capsomeres. So a viral molecule infects a cell normally by docking or attaching to the surface. And this is just a horrible diagram of a viral molecule. I've got the capsid and I've got some genetic material. And if this is the host's membrane, what HIV does is it works to adhere the capsid onto the membrane. So capsid onto the host cell surface membrane, and then it inserts the genetic material, which remember is HIV is RNA. And this goes into the host cell cytoplasm. So if it's us, it's just going to sit and float around as little bits outside of the nucleus, and the rest of the virus remains outside of the cell. So it's only genetic material. I can't be bothered to write genetic material again. So the genetic material inserts into the cell, And remember, we've got those capsid proteins as well. So things that are going to help budding and stuff like that. And finally, when we get inside the capsid, there are a few molecules that are key for HIV structure, activity, formation, and this is comprising of 2 enzymes. So we've got the first enzyme, which is integrase, and as it says there, we are going to input the viral RNA, so the HIV RNA, into the host. And the reverse transcriptase enzyme, which is just in this bubble here, I'm going to label as RT, is an enzyme that turns viral RNA to DNA. And then it can even better integrate into the host's genetic material. Okay, the final thing that we need to know is that in HIV, we've said it's RNA, but it's 2 strands of RNA that can be found in the capsid. Okay, and that's got all of the genes for HIV on them. Perfect, so that's the structure of HIV working outwards to the cell. Okay, so we're going to take a look at this exam question now. Draw and describe the structure of HIV. Now, I know that some of you aren't artists, so you might not want to necessarily draw. I wouldn't want to draw either. So if you don't want to draw, just describe. But if you are drawing, make sure you've labelled it. So you can pause now. You've got about 6 minutes to describe and draw, if you want to, the structure of HIV. All right. So we're back again. We're looking at the answer for this draw and describe the structure of HIV. There's lots of marks that you could have had, and we're going to go through each of those in turn. So if you're thinking about the outermost layer of this HIV molecule where the envelope is, you could say that there's a bilayer made of phospholipids stolen from the host for 1 mark, and for another mark, you could say that there's both host and viral glycoproteins on there. It just says to describe, not to explain anything. So you just need to say what is there. Okay, so you just need to give information. No explanation. Okay, now if it was an explained question, you might say that the host phospholipids, the host glycoproteins, mean that the HIV can evade capture a little bit more. But for this question, that's not really relevant. Working our way inwards slightly more then, outside the capsid, we have got some genetic material. We've got our 2 viral RNA strands, which are inside the capsid. I don't know why that says outside. I'll actually cross that out. Inside capsid. And then we've got viral proteins on the outside of the capsid that are going to help with things like budding off. And finally, inside the capsid, we have got integrase and reverse transcriptase enzymes and viral RNA there, as we've said in the previous bullet point. So each one of those marks that you can see the number 1 in brackets, that's where you could get a mark for. So there's lots and lots of marks available here. 1, 2, 3, 4, 5, 6, 7, and hopefully if you didn't get all 4 of those, then you can add some extra detail to your answer before we move on. So as we've said, HIV is transmitted through physical contact with an infected person, and generally this is the transmission of bodily fluids. We've said already that it's generally likely to be bloodborne, so passed on between like needle stick injuries. So transmission through bodily contact, and we know that it's an infection of T cells, but specifically it's T helper cells. So remember, those T helper cells are those which are going to trigger the B cells to divide, and also to help produce more T cells. So if we wipe out our B cells, both of those are not going to happen. So we get rid of the T helper cells, we get rid of the B cells being produced and the T cells dividing any further. And the way the HIV molecule is going to do this is by using its viral glycoproteins to attach to receptors on the T helper cell membranes. So it's literally just going to dock itself and attach and then insert its genetic material through a process called endocytosis. And that's literally just where the capsid is going to fuse with the membrane and be taken into our host cell. Now for us, that's going to be our T cells. In other organisms, that's going to be T cells as well, because that is the target cell. Remember, receptors are found, or specific receptors are found on specific cell types. So not all cells, will have these glycoprotein receptors. They're probably only found on T cells, which is why HIV is so hard to treat. Now you can pause and freeze this screen, and I'd like you to have a go at sorting these statements out. So putting them in the correct order for me and seeing if you can figure out how HIV regenerates itself or reproduces and then releases itself from the host cell. So pause now. It is a little bit tricky, so take some time, and I guess you could print screen and cut this up as well if you really wanted to. I don't mind. But when you're ready, press play and we'll go through the answers on the next slide. So it was quite a tricky task. The first thing we needed to realise was that nothing is going to happen unless the virus is attached to the host cell. So the virus is going to fuse with the cell's plasma membranes, and the capsid proteins release the viral genome. Remember, that's 2 lots of RNA, and we've got the integrase enzyme, and we've got reverse transcriptase. Okay. Once we're inside of our host cell, the reverse transcriptase, which I'm going to abbreviate to RT from now on, makes DNA from the RNA. So if you look quickly just to the right of this box, if I've got some viral RNA, remember RNA is single-stranded, then what I've got is reverse transcriptase, which I'm going to draw as a little green blob. Reverse transcriptase is going to use all of the nucleotides that are in the host cell to make a strand of DNA. It's made using host nucleotides, and it's going to be complementary to that RNA. It's actually called reverse transcriptase because you are reversing the transcription phase of protein synthesis. So normally when we're making proteins, we'll go from DNA to RNA, but this time we're going from RNA working back to DNA. Now obviously for the third step, we can't integrate the genome of the HIV molecule as a strand of RNA and DNA. That's not going to work. So what instead happens is that strand of DNA that we've made using the host genome, so DNA number 1, and we've got reverse transcriptase, that's going to make another strand of DNA running in the opposite direction, so DNA number 2. And now we've got some double-stranded DNA that contains the viral genome that can be inserted into the host. And that's done in step 4, where we incorporate that viral DNA now into the cell's DNA using integrase. Okay, that enzyme that is going to help integrate. Oh, integrase, not integrate. That's rubbish. Let me rub that bit out again. So using integrase, which integrates it into the cell's DNA. Now, because the gene has the potential to become a virus, we're going to call it a pro-viral gene. So I'll write that down here, potential to become a virus. Okay, and those pro-viral genes that we spent all of that time going from HIV RNA to one strand of DNA to make another strand of DNA, we're now going to make back into RNA molecules. Okay, those RNA molecules are going from DNA to RNA, and we know that the next step is taking that RNA and transcribing it from RNA to proteins. Okay, so we're taking RNA to proteins, and these proteins are going to be parts of the HIV structure. So this is where we're making lots of new HIV molecules. Now remember, our HIV itself doesn't have any ribosomes, it doesn't have any Golgi apparatus or anything like that. So it relies on integrating its genetic material into the host in order for these proteins to be made in order to make more HIV molecules. And what we get finally is capsid proteins, or capsids using capsomeres, forming around those viral genomes, that RNA that we've made, the HIV proteins, including enzymes. Remember, reverse transcriptase and integrase are enzymes, and enzymes are proteins, and they are going to be made through the host's cellular components. And then finally what we've got is those new cells, the capsids, with all of the genetic material being budded off from the host cell to integrate some host glycoproteins into its membrane, into its envelope. Host glycoproteins into membrane, or envelope is probably better. Okay, so once you've thought about it like that, it's actually quite a commonsensical process. It's just a little bit of a wordy time to get your head around. Now, as we've said previously, after you've been infected with HIV, you have a period where you get more and more HIV being produced. This latency period, there's more and more HIV being produced, but you don't have symptoms. And that's because it takes a certain viral load. Okay, and viral load is a really important term to be able to say about. A certain viral load to elicit the symptoms. And this would be where you've got few T cells, or so few T cells that you're really susceptible to infection. Susceptible to infection. And this is the time where you'll feel tired, lethargic, you'll get colds all the time, pneumonia, things like that, and you'll be like, oh, something's actually really wrong. Now the symptoms are going to occur when the host cells are starting to be taken over, they're overrun by HIV. And at this point, the T helper cells are being destroyed as that HIV begins budding off the cells. So all the time, the HIV is just multiplying inside the cells. So HIV multiplying, but not budding. Then we don't get symptoms. But as soon as you've got those HIV molecules starting to bust out of those host cells, or budding off, which is probably the more scientific way to say busting out, you're going to start getting those symptoms coming through like wildfire. And this is because you've got a significantly weakened immune response. Okay? And saying that it's significantly weakened is key. We've got a reduced number of T helper cells. Don't forget the T helper. We have to be specific. So knowing what we know now about the structure of HIV, how it replicates, things like that, have a go at doing some research, and you can pause me for a second and find out how reverse transcriptase inhibitors and protease inhibitors work. They're both HIV treatments, and you need to find out and think about how they can be used to treat HIV. So pause now, and when you're ready to go through the answers, play again. Now hopefully you should have been able to recognise that reverse transcriptase inhibitors are just going to work by stopping viral RNA being copied into those two DNA strands at the very beginning of the HIV life cycle. In doing that, we stop the DNA being available to integrate into the host, and therefore stop the formation of any more viral molecules. In terms of protease inhibitors, we're stopping the use of proteases, those enzymes that are going to break down proteins and stop being able to produce new viral proteins. So if we can't make new viral proteins, then we are not going to be able to produce those new molecules of the virus, and therefore they're not going to be able to do the budding off and release from the host cell. Now, we need to think about how the HIV life cycle actually means that it's really, really hard to treat. And we've got to think about it in these terms. So HIV steals host glycoproteins from the plasma membrane as it replicates, specifically as it buds off. Why is this an issue when considering treatment options for HIV? So you can pause, you can have a think, or you can just carry on if you want to find out the answer. So cells infected with HIV look normal. Okay, remember, they're just going to have their normal cell structure, and inside them is where there's lots of HIV. Inside lots of HIV. Okay, the viral structures themselves, so the HIV molecules, steal those glycoproteins. And in doing the stealing of the glycoproteins, glycoprots, then they're going to be able to evade that capture. Now, both of these are slightly problematic, because if an infected cell and a HIV molecule looks like a self cell, so it looks like a self, then it's really difficult to target. And we don't want to target our cells that look like our body cells, because otherwise we'd be destroying lots of tissue around them. We've also got another issue in the fact that HIV also mutates. So when cells replicate, normally there's some form of mutation that goes on, and normally if the mutations are bad, especially in our body cells, for example, then our body would just remove those cells and let them undergo apoptosis, that programmed cell death. However, in HIV, these mutation processes give rise to randomly resistant viruses. And these randomly resistant viruses are actually resistant to the anti-HIV medication. And this in turn means that sometimes some of the current treatments are ineffective. And we're going to take a look at how that works actually in a second. So normally, when an individual is being treated for HIV, they're undergoing HIV treatment therapies and things like that. We're going to have to use lots of different medications. It's normally a combination of 3. So if we have a look here at what happens if you're only taking one treatment, here in the left-hand diagram, I've got lots of HIV molecules. All of those circles are HIV. The grey ones are the wild type or the normal HIV, and the 3 different colours show resistant types, and you can see that down in the key at the bottom. Now, if I give a treatment of drug 1, the drug 1, as it says down here, works against the wild type virus, that normal virus, and R2 and R3. So over time, R2 and R3 are all going to get knocked out or killed anyway, and so are all of the wild type ones. But you can see over time, R1 isn't being destroyed, and that means that R1 virus, that R1 type of the virus, is able to multiply lots and lots and lots and lots of times over, and you're still infected with HIV. Okay, you've still got that virus floating around in your body. You're now infected with a different and more resistant type. So if anything, despite starting treatment, in this case, you've made it a little bit worse for yourself because the drug isn't having an effect. Okay, so if we give an individual 3 treatments, which we'll see out on the next slide, it's slightly better in terms of long-term treatment options. So here I've got my HIV blood sample here. I'm HIV positive. I've got the wild type virus. I've got R1, R2 and R3, which are all different mutations of that HIV virus. And what I've done this time is I've given 3 different drugs. And as you can see in this slide, or in the next picture along, each of those drugs works and is specific for a few different types of those HIV. So those drugs are active. This combination therapy means that over time, because of that combination of each of the drugs affecting different HIV subtypes, then we end up with a lot less HIV. Less than 50 copies per milliliter is a low viral load or an undetectable viral load. Okay, and this is where it's very unlikely that you're going to pass on that HIV because you've got such few molecules of it floating around in your blood. Okay, so if we skip back and forward again, if you go back to the previous video, you saw that after that single drug treatment, there was still loads and loads of viral molecules, but in this combination therapy, there is no or very, very few viral molecules left. Now this says homework. You could do it as a little research task if you wanted to, because it does come up quite a lot in the stretch and challenge questions if you're looking for an A star or an A. What is AIDS and how would a doctor differentiate between HIV and AIDS? You might also want to consider how a doctor could diagnose AIDS. Okay, what are the key clinical symptoms for AIDS that are different to HIV? And to end up with, we always finish on an exam question. So this one is from June 2016. Table 1 shows a feature of a bacterium and the human immunodeficiency virus, or HIV. Complete Table 1 by putting a tick where the feature is present. So I would suggest that for 2 marks, you spend 3 minutes on this. You can draw the table out, or you can just, I guess, screenshot this and insert it into your OneNote area for your notes for today's lesson. But have a go, and then I'm just going to be quiet for a second, and I'm just going to move on to the next slide. So pause now. Okay, this is potentially a little bit more of a wordy one. So when HIV infects a human cell, the following events occur. A single-stranded length of HIV DNA is made, and the human cell then makes a complementary strand to the HIV DNA. The complementary strand is made in the same way as a new complementary strand is made during semi-conservative replication of human DNA. Describe how the complementary strand of HIV DNA is made. Now this is where we're bringing back module 1. Okay, because it's effectively asking you, how does semi-conservative replication happen, and how does it make a double-stranded, how does it make another strand of DNA to make that HIV DNA double-stranded? So it's 3 marks. You've got 5 minutes. Give me a pause, and then when you're ready, we'll go to the answers, and then we are done. So structurally, both the HIV and a bacterium have RNA. Good. The cell wall is only found on a bacterium. Remember, in the HIV, it's called the envelope. In both bacteria and HIV, we've got enzymes. We know that in HIV, they're reverse transcriptase and integrase. And finally, the HIV molecule itself has that capsid. We've not got a lot of leeway in the marks here because it's 1 mark for each correct column. So you had to get all of those right and all of those right to be able to pick up 2 marks. Otherwise, you got none. So if we carry on down here, it's talking about that process of semi-conservative replication. So what we should have been able to say at some point in our answer is that the nucleotides or the bases pair complementarily, or the A goes to T and C goes to G. You cannot just mention A goes with T as an example because there's 4 nucleotides. You need to mention both of them if you're going to mention them at all. If you've got any mention of DNA polymerase, then that's another mark. And the final mark is for saying that the nucleotides join together to form the new strands, or phosphodiester bonds form between the sugar phosphate backbone, for example, where we've got that deoxyribose and the phosphate molecule. Now, it does say that if you are clearly writing about DNA replication and not this semi-conservative replication, so maybe you've said about the fact that the DNA strands will separate, helicase will work, something like that, and you're visibly not understanding that we're taking a single strand of DNA and just building onto it, then you can only get 2 marks maximum. So I hope you found this useful. If you've got any questions about HIV, then I guess you can email me, pop up on chat, ask the question. Otherwise, this is the end of Module 2. So for the next lesson, we're going to be producing one of those big mind maps that encompasses all of the topic, all of together, and then we'll have some revision time before your Module 2 assessment. Thanks very much, guys. I'll see you later. Bye-bye.